Hey everyone, um, welcome today. If you have any questions as we go along, please don't hesitate to raise your hand or unmute and stop me and we can discuss. I am the operations manager for IU-Bloomington. Karen Rogers, our attending veterinarian, was unable to make it today. So the mission of our laboratory animal resources group is to provide high-quality, humane care for research and teaching animals on Indiana University Bloomington campus and ensure compliance with federal laws, guidelines, campus policies related to animal use. We are responsible for managing the campus core animal facilities and providing veterinary medical care and oversight for all campus facilities in which animals are maintained for teaching and research. So a quick overview of our program. We do have 6 core animal facilities across campus. They cover several departments and areas and lots of research, including but not limited to cognitive neuroscience, cellular and molecular neuroscience, development disorders of the nervous system, homeostatic and neuroendocrine systems, sensory and motor processes, infectious disease. We currently have a total of 14 staff members, so 2 full-time faculty and 12 full-time staff. And we house currently a variety of species, including birds, hamsters, rats, mice, fish, and frogs. There is regulatory oversight of our program by the BIACUC, so the Bloomington Institutional Animal Care and Use Committee, the USDA, and ALAC, so the Association for the Assessment and Accreditation of Laboratory Animal Care. So this just gives an example of one of our core facilities. MSB2 Animal Facility is located on the ground floor of the MSB2 building. And it is associated with the Gill Center for Neuroscience. We do support several of those investigators. So our primary responsibilities include husbandry, so the daily care. The daily care for the animals, administration, making sure that everything is being processed. We have supplies that are needed on hand, veterinary medical care, and compliance. So to take a look at some more of our husbandry responsibilities, changing the cages, making sure that the animals have food, water, bedding, making sure that our rooms are clean and sanitized, maintaining our equipment, making sure that our facilities are stocked. Some more of the species that we take care of. So we have some frogs, some zebrafish. Several of these species are used for developmental type studies, but their racks and their care also have some specialized, so though we're not changing bedding, we are making sure that they have water quality and making sure they're being maintained and cared for. And then making sure that we're keeping everything clean and processed. So the staff daily, as soon after checks are done, will go through, they'll dump cages, they'll wash their cages through cage wash, cage racks. The same thing as kind of a large dishwasher. And then make sure that they're emptying those, refilling them, and getting them ready for the next day. From an administration standpoint, we do manage many of the facility operations. So training, HR, hiring, performance management of staff, making sure that we get animal orders in, processing the animal orders, processing animal transfers, billing for per diems, the animal census and tracking, inventory, making sure that we have all the supplies that are necessary, the feed, the bedding, PPE, to make sure that everything can run smoothly, helping to be the communication between facility operations and our investigators when construction projects are happening or renovations or space renovations need to be made. And then also transporting animals across campus to labs, between facilities, so that we can best utilize all the spaces that we have on campus. We do have 2 veterinarians on staff. So our attending veterinarian is Dr. Karen Rogers, and our clinical veterinarian is Dr. Randolyn Shepard. So veterinary, we also have 1 animal health technician, Caitlin Hossam. But they diagnose, treat, monitor the animal care. They also consult on colony management, surgeries, euthanasia, looking at the pathology, disease progression. They also help our staff to evaluate the environments of the animals, to make sure that we're maintaining and everything for the animal welfare side. Here is a picture of one of our procedure rooms in the MSB2 building and a rodent surgery suite within that space. Both of these rooms are rooms that we have available. They're through our core, but we do sign up. We have schedules and reservations so that they can be utilized. And then compliance. I mentioned earlier that we do have regulatory oversight by the USDA, our Institutional Animal Care and Use Committee, and ALAC. Our veterinarians also consult on several protocols, models, the animal models, and helping with the protocols, justifying the protocols, making sure that we're doing post-approval monitoring to make sure that we are upholding that level of animal care. The compliance aspect of making sure that we are obtaining our high standard of animal care. So to kind of summarize everything that we've discussed, our functions and services. We do provide the animal ordering, animal receiving, and billing, the housing, space, and husbandry services for the variety of species, clinical veterinary care program for all the research animals on campus, as well as the health surveillance program and pathogen monitoring, the ordering and provision of food, water, and housing, veterinary consultation and training on animal care, handling, dosing, sampling, and surgical methods, the assistance with protocol writing and addressing IACUC questions, training and education on animal handling or research issues, collaboration on research projects, determination of housing space for animal researchers, provision of some equipment for shared use by PIs, ordering of drugs and materials requiring a veterinary license to purchase, and diagnostic necropsy and histopathology on animals with spontaneous diseases or study-related side effects. We do offer some services for additional fees. So shipping and receiving of animals from other research institutions, so non-routine vendors. We do have import-export charges for coordinating those shipments. Services such as administering special feed or dose solutions in water, weakened treatments. At the beginning of the pandemic, we did have to or during the pandemic, we did have to get away from breeding colony management services, but we hope to be able to get back to those again at some point. And then separation of overcrowded cages. So I mentioned in the beginning with the compliance that we do have regulatory oversight by the Bloomington Animal Care and Use Committee. Their website is listed here, but the IACUC is, the goal is that they review work proposed on animals, and it must be submitted in a protocol form to the IACUC and approved before work begins. So modification to animal work must be reviewed and approved by the IACUC before it's implemented. The IACUC does perform semi-annual inspections of the facilities and of the labs. The animal facilities and spaces must be inspected and approved by the IACUC prior to animal use beginning within these areas to make sure that it's safe and within guidelines. And then the IACUC issues and maintains policies related to animal use that need to be followed by the research groups. And again, there is a more comprehensive list of all of these policies located on their website. LAR also has an advisory committee that meets 2 to 3 times a year at a minimum to discuss financial issues and then other issues concerning the LAR department. The chair for this committee is Dr. Ken Mackey, and we try to have a faculty member from each of the different departments that we serve serving on that committee. So research planning. This is kind of where we need our customers to help us. So we need to know when there are new animal users coming, when either if that is a new hire. I know there's lots of hiring going on right now. So new hire, new animal users for Bloomington campus, or established researchers that are going to be new to working with animals. So they've been here a while, but they have not moved into animal research. So it helps for us to know the species of the animals that they're going to be working with, the number, the housing type, to help determine the appropriate housing space and any type of special needs services or supplies. Something to keep in mind is that unfortunately we do have a finite amount of caging supplies. And so when we start adding new people to this, we do have discussions with departments and research administration about receiving finances to purchase additional supplies to support these projects. We also ask that departments keep us updated on animal needs and if new grants are obtained and if space needs change to kind of help us plan ahead to know where we're going. And with all the new hires, we really like for our attending veterinarian to be included on these interview schedules to tour the animal facilities and start those discussions very early on. The more time we have, the better prepared we can be. So hiring of new faculty animal users. When considering to hire new faculty that use animals, again, please include us in the hiring process so that we can work with the faculty to appropriately house their animals and meet their needs. Candidates should not be hired with the promise of having animal space at the university without first discussing the needs with us. We have had a couple of investigators that have showed up and just asked where their animal space was. And Dr. Rogers unfortunately had to say, "Who are you?" So we like to avoid that. So again, include us as soon as possible. Once animal facility needs are known and shared, LAR can proceed with providing information about housing for animals. Final decisions on space and housing will need to be negotiated between the hiring department, the dean, and LAR and OVPR, preferably before a hiring decision is made. So a couple of things that we like to ask of new employees or new hires is a little bit about their research, the species that they're going to be working with, their approximate colony size, the cage types, if they're going to have any type of special behavioral equipment or imaging equipment that they will need, what the procedures will be performed, what kinds of procedure spaces that will be needed, and anything else that might be included in that. Are there any questions? We do have a website, so feel free to visit our website or also contact us.
All right. Thanks, Carrie. Great job. Thanks, Jill, for the invitation here. So I'm going to talk a little bit more about a specific piece of equipment that's located within LAR that Carrie also helps manage and is part of their program here on the Bloomington campus. So I'm a faculty in the School of Medicine here on the Bloomington campus, and sort of helping manage this equipment, the IVIS Lumina system. I spent a lot of work trying to get the system here to the Bloomington campus because we had one prior to this one that was about 15 years old. So we've sort of updated to a newer system. So I'm kind of excited to talk to people about this and hopefully get some more users here on the Bloomington campus. So what are we talking about here? The IVIS Lumina system. So these are imaging systems for in vivo imaging of animals, small animals, and they're the company that makes them, PerkinElmer. They have a range of these different systems. So you can see different ones here on the left that are benchtop systems that look sort of like a small mini refrigerator that can sit on the benchtop. And then ones over here on the right are larger ones that look more like an actual refrigerator you would see in your home sitting on the floor. So here on the Bloomington campus, we have this IVIS Lumina X5 system. If you are on the Indianapolis campus, they do have an IVIS Spectrum CT system that is also capable of doing all of the things that I'm going to show you today that the Lumina X5 can do as well. So this IVIS Lumina X5, I'll go through some more of these details so you don't need to try and read this table. But this is kind of what it looks like. And we can open the doors and put in our animals into this. And then we have a camera that sits up here at the top. Let me turn on my laser pointer. That sits up here at the top that can read and take the information and the data that it's collecting from the animals that sit here on this stage. It also has X-ray capabilities down here on the bottom that go upwards. So what we can do, as I mentioned, is a bioluminescence imaging. So this is going to be imaging of bioluminescence, which I'll talk briefly about more momentarily. So it can do 2D bioluminescence imaging from this camera up here at the top. And so it can read this data in the pictured here as a mouse, but it can do other small animals as well, or even cells in culture. It can also, so you can see this is what that looks like in a real mouse, where we have, in this particular case, we have a tumor growing that's been labeled bioluminescently, so we can actually see that tumor in the mouse. It can also do fluorescence. So it has a light source on the side here with different excitations filters that I'll mention momentarily, as well as an emission filter, and then our camera that can collect the data at the top. And we can also get these type of similar images if whatever your material is labeled with a fluorescent marker instead of a bioluminescent marker. And we can also do, as I mentioned, a low-dose X-ray or micro CT. And so that is coming from the bottom of this chamber. And that can largely give us structural information about the animal that you're imaging, which you can see here in this particular case is a mouse. So if you're interested in any aspects of the structure of the mouse, which I'll give a couple of examples later where people have used this, this could be of use to you. So very briefly, what is bioluminescence? So this is just source of biological species able to produce light. And so this is not necessarily fluorescence, just light, so luminescence. And so this is typically done by animals that have an enzyme called a luciferase enzyme, which the example here is for the firefly, where it can take this luciferin substrate, and this enzyme will convert that to this oxyluciferin, and then more importantly, light. And that is what we see when we look at fireflies and they're glowing here in the summer. And we can use this a lot in biological systems where we can actually use the gene for this luciferase and put it into different reporter systems that can be driven to test and study whatever it is that you'd be interested in studying. So in this particular example here, this is a reporter that has the luciferase gene tied to a very specific promoter that reports the activity of a specific transcription factor. So anytime we see the amount of light that the lysate that this system is in, we know how much activity of our transcription factor is in this particular case. For the system related to fluorescence, this table here on the left includes all of the excitation and emission filters that are included with the system. So excitation from 420 up to 780 wavelengths, and emissions from 520 up to 845. And you can see on the diagram here on the right, some pretty commonly used fluorescent markers for this. So on the left here, this is the green or GFP, if you're familiar with these, with the M cherry being here, and then we have a 680 and 750 for some of the further right ones. So this system can image any of these, and there are capabilities for stuff even in between. And so later, if you have interesting or have questions and are interested in using the fluorescence aspect of this, we can always go in and test and see if it's going to be able to distinguish or differentiate between whatever markers you'd like to use. I'm going to show now just a few examples of how folks, myself included, have used this system to get data both for publications as well as preliminary data for grants, and I'll have maybe a couple more comments on that towards the end. So one of the ways that we in my lab use this system a lot is we will engineer our cancer cells to constitutively or constantly express a luciferase gene so that we can then put those into mice and we can track those cells throughout the mice. So to do that, we have to first engineer our cells. So for us in our cancer cells, we would typically infect them with a virus that encodes the luciferase gene. We'll do some drug selection and potentially other types of selection to get the cell mixture that we want. And then we will take those cells that have been engineered to express the luciferase gene, and we will confirm that in vitro first, so we can actually image with our system cells in a 96-well plate like you see here. We just need to add the substrate for the enzyme, which is this luciferin substrate. And then if that works and once we're happy with the cells from the in vitro conformation, we will do the same thing in vivo, where we inject our cells into mice, and then we can inject our substrate D-luciferin into the mice, usually through IP injection. And then that luciferin will circulate around the mice, interact with these cells, and those cells will convert that D-luciferin to light, which we can pick up in this IVIS system. So this is one example of how we commonly do it in my lab, but there's plenty of other ways that you can do this. So you could do the exact same system actually, where you just have a fluorescent label instead of a bioluminescent label. Other folks have used this to label bacterial cells, which they can put into animals and then track the infection after that. Similarly, you can engineer viral particles to include as part of the virus some type of reporter, either fluorescent or bioluminescent reporter, and then infect your animals. And then you can track that infection over time. And we'll see a few examples of these things momentarily. Other folks have even used nanoparticles, where you can load in things like drugs for treatment But you can also load in markers such as fluorescent markers and even bioluminescent markers with those nanoparticles and deliver those in vivo and then track those. And similarly, Micelles, which are sort of lipid-contained systems where you can also include either a bioluminescent enzyme or a fluorescent marker as well. And then there are other folks that have also engineered genetic models to have certain cell populations to have either a bioluminescent or fluorescent marker, so that if you image the mice at any one time, you would be able to see where that cell population is in the animal at any given time. So a few examples of this. So in our case, we will typically use something like this to look at things like drug response. So this is one of the pathways that we have looked at, where we have combined these two drugs as a therapeutic treatment, and that we can track these mice where we have injected tumors into their mammary fat pad to model breast cancer. And then we can track them over time, day 21, 28, 35, and we can see that these tumors are growing bigger over time, and that our combination treatment is more effective at preventing that growth than some of either the single treatments and compared to this vehicle treatment. So it's a way where we can actually see the tumor population here getting smaller or not getting bigger in this particular case. We can also use it to track cancer cells. So similarly, we can study the same pathway. And if we take our cancer cells, we can actually directly inject them into the left ventricle of our mice, and that heart then will pump and shoot those cells throughout the circulation system. Those cells can metastasize and grow metastatic tumors throughout the body. And so we can see that on the mice here on the left, where we can see this. All of these shaded areas are different and unique metastatic tumors that have grown and established in these mice. And we can see that if we get rid of one component of our pathway here, that we can significantly reduce that metastatic burden that we see on the mice. So it's a really nice way to track how these cells are spreading throughout the animal without having to actually sacrifice the animal. So we can actually image these longitudinally over time and track how this same mouse here is progressing over time, and how the tumors within that mouse are progressing over time. So similarly, as I mentioned before, we can look at things like drug distribution. So this is an example where we have taken, this is not my group actually, another group has taken Micelles and loaded them with this compound here that has a fluorescent marker in it. And then they will inject that Micelles into mice. And so you can see immediately after the injection that you can see the very broad distribution of these Micelles in the mice, and that over time, some of them are getting distributed more specifically to more specific tissues. And so you can see in this particular case, they're looking at delivery to a tumor. So you can see the delivery ends up staying fairly specific to the tumor. And that at the end of this study, they actually ex vivo excised all of the organs and then imaged the organs ex vivo. And you can still see what the distribution of those Micelles are in these different organs here, which we can then use the analysis software to quantify the fluorescence in this case of reporting back the levels of Micelles in these different tissues. So we can actually see what the distribution is, and if we're getting, in this case, specific distribution to the tumor, which was, I believe, the hope of the study. Other examples, so there's a very interesting one using a bacterial distribution, or trying to study bacterial distribution and how that bacteria is having an effect on bone damage. So what this group used is a Staph aureus strain that has been engineered to express this luciferase enzyme. And they will infect these mice here with the Staph aureus. And these mice are genetically engineered mice to express a GFP, a fluorescent marker, specifically in their myeloid lineage of cells. And they're specifically interested in how this Staph aureus is affecting the bone damage. So they would infect the mice with the Staph aureus, particularly in the bone regions. And then they will use the bioluminescence to detect the levels of infection of the Staph aureus. They use fluorescence to look at the levels of the myeloid cells, which in their case, because of a more of an inflammatory response, this is going to be mostly neutrophils. And then they can also use the micro CT function of the system to look at what is the actual bone damage happening and get some quantifications of that. And so what the data sort of looks like over here from one of the figures from their paper, you can see in A, they have the bioluminescence that's reporting that the infection actually happened and how long the infection was maintained. And then in B, they can see what is the particular neutrophil or myeloid cell levels in this particular area of the bone, which you can see imaged here in C, and what that looks like over time. And then C is more of an image of the actual bioluminescence in the top here, which you can see where the infection is and how it's changing over time. And then in the lower one here is the GFP marker, so this fluorescent marker that you can also see is changing over time as the infection is starting to wane. So this is a very interesting study where we essentially used every major function of the system in one study, and we're actually able to get all of these pieces of information from the exact same mouse, both in terms of getting the bioluminescence and the fluorescence, but also over time, this 2, 5, 14, 19, 28 days, and so on. This is an image from the exact same mouse. So we're not dependent on using groups of mice and getting averages, although you can certainly get that. But we can actually track these things in the exact same mouse over time. Similarly, for bacterial distribution, I know on the Bloomington campus, we have a lot of model systems here, as Carrie described very well, including things like fish and other species. So here's a group that actually used fluorescently labeled version of an E. coli strain, and we're infecting fish and then using IVIS imaging to detect what the infection was looking like in those fish. And so you can see here in A is, their in vitro data where they can see that the E. coli is actually fluorescing and have data clear on that. And then C here is the images of the actual catfish in this case that they were infecting with the E. coli, and they could see what this infection looked like both over time and in response to some other things that they were doing to the fish as well. So similar to bacteria, as I said, we can also do this with viruses. So viruses in this particular study, you were studying how COVID infection was affected by some of the neutralizing antibodies. And so they engineered the COVID virus to express this small version of a luciferase gene. And then they were taking this COVID strain here and then infecting mice nasally in this case, and then taking those mice and doing bioluminescent imaging, and they could track the infection over time. And in this particular case, they were interested in how some of the neutralizing antibodies were affecting the infection. And we're actually able to get some really information about what are some key components of that neutralizing antibody to help protect against the infection. So they're able to get whole animal imaging here longitudinally over time. And then also at the very end of the study, when they euthanize the mice and open them up for necropsy, they also do another final imaging here. And then they will also excise all of the organs that you can see here, and then do bioluminescent imaging on those to get quantifications of the amount of infection in each separate organ. And so we're able to use this for a lot of things in vivo, everything from cancer cells to any other mammalian cell that you would be interested in, to bacteria, to viruses, anything where you would really want to track any particular species traveling throughout another species, another organism. So we can also, there's also been a couple of uses of this ex vivo. There are probably slightly better systems than the IVIS to use for some of these ex vivo uses, but people have used it. So on the left here is an example where we have taken cells and engineered them to express specific genes with a Firefly reporter. And then we can actually do drug treatment on those to see which drugs were preventing the activity of one of these genes versus the other, and actually identify drugs and targets that way. Similarly, because we are able to detect light and luminescence, people have also used this to get very highly specific quantifiable data from things as simple as Western blots or immunoblots, where you can see the IVIS imaging of these bands here. And then we can use the software to do a very good analysis and get very quantifiable data from an otherwise fairly semi-quantitative method. So there are other opportunities for things like this that you can also use it for. As I mentioned, maybe some of these now might have slightly better techniques or better technologies, but it is here to use for things like this or any perhaps new ideas that you would come across. So the system does come with its own software, analysis software. It's called Living Image. And we do have that here on the Bloomington campus. And we have access to a few different ports for analysis. So we don't need to actually only use the system, the software that's controlling the actual imaging system. So we do have some other ports that have the software available for people using the system to do their analysis. And so you can talk to me about that later if you're interested in using this, and I can point you to where those are. But the software is very good where you can get quantifiable data. In this particular case, we're looking at mice, where you can get whole body data if you want to enclose the entire animal, if you're interested in the whole body fluorescence or bioluminescence, or you can make specific regions of interest to get much more specific data depending on really what is your research question. So there's a lot of opportunities for good analysis, and the software is very good at maintaining balances and normalizations across samples and across longitudinal time points, which is something that some of the older versions of the software that I've used in years past were not very good at, but the newer version that we have available is actually very good at doing that and saves us a lot of manual work in doing the analysis for this. So I think probably one of the biggest perhaps benefits of this type of system is doing longitudinal studies. So in previous times, if we wanted to do one of our metastasis studies that I described earlier, we would have to take our mice, give them tumors, and then as weeks go along, we would have to take mice out of this population and sacrifice them, and then look at the tissues histologically to see if we actually see metastasis or not. So over time, our mouse population starts to dwindle until we see our metastasis or our endpoint of interest. And so typically we would have to compensate for this loss of animals over time by starting with a much larger animal population at the beginning of the study. However, using something like this non-invasive tracking that the IVIS offers, we can take our mouse population, give them tumors, and then every week, or even sometimes twice a week, we can image these mice, and we don't ever have to sacrifice these mice just to see what is the status of our tumors in our study. So we don't have to compensate for buying extra animals. So it saves us a lot of money in terms of animal costs. And additionally, we can then also track progress in the same animal over time, which can be useful depending on what your research question is. Sometimes we just want to know what the average or the mean or median of our mouse population is doing. Other times we want to see what are some of the specific differences in heterogeneity within the population, and see what any individual animal is responding to either our tumors, to a bacteria, to a virus, or whatever it is that you would be studying. And so it has a lot of benefits for this, and probably, as an investigator, the biggest benefit is going to be cost savings. So it's very helpful in that regard. My email is here at the top that you can email me and ask me for questions or help with this equipment later. The IVIS Lumina system here on the Bloomington campus is located on the biology building and the animal facility there. If you are on the Bloomington campus and have animals at one of the other sites, LAR does offer transport of animals between sites on the campus. That's something you can ask Carrie about and see what that service requires and what you need to put in your animal protocol in order to make that happen. If you're interested in using the equipment here on the Bloomington campus, you can go to iLab, where LAR now is providing their services, and there's a request training for the IVIS on that. So if you click that, they will get a message to me, and then we will get in contact about doing a training an in-person training on how to use the system. And you will have to be an approved animal user on an approved IACUC protocol in order to use the system for in vivo use. If you want to use it for in vitro use, you can also reach out to me and we can set you up to do that as well. And then something else that is quite useful is that the LAR is a CTSI core facility, so use of the IVIS and even other LAR services are eligible for the CTSI core pilot grants. So this is a really great way to use some of that money to help further maybe some of your preliminary data for future grants. And if you have any questions about the system, about how to use it, about how to design your experiments, how to maybe engineer your systems to make it usable for this, for the IVIS, I'm always here to help. So you can reach out to me by email. And I'll be happy to walk you through that. And that includes help with and designing very small studies that I would typically call feasibility experiments that you can be used as preliminary data for your grant proposals. So if you just need, one figure to show that your system does work in theory, even if it's only one animal that you've gotten it to work so far, that's feasibility data that you can use for your grant proposals. And so I'd be more than happy to help you set that up and help you use the system and help you design your reporters and your other parts of the system in order to get that feasibility data, and then hopefully later use your awarded grants to come back and use the system for your bigger studies to get publications and so forth. So that's all the slides I have. I'm more than happy to take any questions or talk about any part of this that you guys would be interested in.
I guess my question is for anyone who is like us, who don't do this kind of work, but hear about your kind of talk and say, oh man, I would wish I could do that because I would write a nice grant or whatever based on this technology. What services can LAR offer beyond for the newbies like us, or people who don't do this kind of work but see the importance of it? Can you speak to that a little bit more? I know you guys do a service, a husbandry and all of that stuff, and you do offer, like this technology, but I'm wondering about people who might be on this call right now, who are like myself, don't have any experience in this, but would like to talk to someone or maybe set up a mini collaboration potentially. Is that in your in your sites, or do you come?
Yeah, so I can have a few comments on that, and then I'll let Carrie kind of add the probably more official comments. Yeah, so I think the path for you most likely, right, is to find a collaborator, someone who is familiar with whatever organism you want to actually infect later. So if it's mice, preferably someone who's worked with mice. And I'm happy to talk to you more about this later and help you find the right person. And probably it's to do, give you guys work together to get the infection in that organism and see if you can detect it with a system like this. And if you can, then it's probably just a matter of ramping up and getting the right personnel to actually perform the experiment, right? And yeah, I'm happy to facilitate as much of that as possible. And if it's just a matter of going and imaging the mice, there's plenty of us that know how to do that, who don't have experience with the bacteria that you're talking about or the nanoparticles. So you'd have to have someone be there to do the delivery of that and probably, help with the interpretation of the analysis. But so in terms of collaboration, I'm certainly can help you with that. And it can even look a bit more into your markers and stuff and see if they're going to be, work with the system, which I'm sure they will be. And really help you make sure that it's going to work for you and get it to work for you. And then I can also probably help facilitate finding the right person to be your collaborator for that. And I think, I'll very briefly say unofficially, I think we had previously talked about LAR potentially in the future offering services of imaging, but that is, to my understanding, not currently available. So it would have to be a collaboration of.
Does anyone else have a question? Okay, well, if not, I would like to thank Carrie and Richard again for presenting. It was a really nice overview of the Animal Resource Facility in Bloomington. And so, all right, thank you.